Our next speaker is Erin Levesque, who is the fishery biologist at the Marine Resources Division of South Carolina DNR and is responsible for managing our local Waddell Mariculture Center. And she will be talking about food web lessons from mariculture of red drum.
Good afternoon, everybody. Can everybody see my screen?
Yes.
Okay, perfect. So thanks for inviting me today. I'm actually a part of the estuary and finfish section. So the same larger section that Joey and Elizabeth are a part of, but more specifically, I am a member of the mariculture section. And our mariculture section is responsible for the production and culture of marine and estuarine finfish, particularly those that are recreationally important that Joey was speaking of. And our production ponds, you can see here in the foreground. So our production schedule in our ponds operates typically from around April to November. We're culturing currently in our ponds cobia and red drum. I'm going to be specifically speaking to red drum in this talk and temperature and prey constraints on our pond production of Duvedal red drum. You can see, I want to point out in the background here, the Colleton River. So this is where our property sits, and this is important because this is the water source for not only all of our pond production, but also all of our hatchery production here at Waddell. So you can see an aerial shot here of our production ponds. For production, we're really primarily using these 3 large ponds. These are 1.25 acres in area, and our medium-sized ponds, which are just a little over a half acre. They're essentially acting as mesocosms, and what I mean by that is, in addition to bringing in water from the Colleton River, we're also bringing in all of the organisms that inhabit that water, specifically phytoplankters. So these are microscopic plants, and zooplankters, microscopic animals. And these are extremely important because they form the basis of our food chain in our ponds. So when we are stocking our ponds with larval red drum, the zooplankters, these microscopic animals, are the first prey that these small fish are going to encounter. And in fact, they're the same organisms that they would encounter in the wild in Port Royal Sound estuary. It takes us about 7 to 12 days, depending on the size of the ponds, to get them completely filled. And at that point, we turn the water off. So it becomes a closed system. So a little bit different than the estuary. They're not experiencing tidal flows daily. We're also limiting who comes into our ponds. So we are introducing only small phytoplankton and zooplankton. We are excluding any larger fish larvae or fish eggs, anything that can potentially be another predator in the ponds or a competitor to our red drum larvae. So although the ponds are functionally a bit different than the estuaries, we can use what we learn in the pond system and those dynamics between physical and biological mechanisms in order to learn about what's happening with red drum year classes in the wild. So just to compare some other aspects of ponds, our pond production to estuaries. So again, the water source is the same. We're using Colleton River water. However, there's a huge difference in volume, right? So our ponds are smaller in area, they're shallower, and they're not experiencing tidal influx. And again, they are closed systems after they're completely filled. But as a result of that, what we're going to see are pretty large daily fluctuations in water quality parameters like water temperature and dissolved oxygen specifically. So you'll see some of these daily fluctuations as well in the estuary as a part of just natural biological processes. They're just going to be more pronounced in our ponds. So for example, the phytoplankton in our ponds are photosynthesizing during the day. So we're going to see the highest dissolved oxygen levels in the afternoon when most oxygen is being produced. It's been accumulating all day long. However, at night when sunlight isn't available, those phytoplankters switch to a respiration cycle, as well as all of the zooplankton and the fish in our ponds are also respiring, consuming the oxygen. We're going to see the lowest levels in the morning, just before sunrise. So I mentioned we are controlling somewhat who comes into our ponds, the organisms that come into our ponds. So we're really looking at more of a food chain, so a very distinct unit in terms of predator-prey mechanisms, whereas in the estuary proper, you're going to see these food chains interconnected and forming food webs. And both systems certainly experience various challenges. So we are able, because we're condensing phytoplankton and zooplankton in our systems, we're actually able to see a pretty high density of prey items in our ponds, which is great for growth and survival. However, because we are a closed system, as the fish begin to grow and consume more prey, we're going to see that prey base significantly decrease. Whereas in the estuary, you'll see patchy resources, so not quite as dense as we have available in our ponds, but they're replenished because of volume and because of tides. So this is an example of the food chain that we would see in our ponds. So up here in the top right, you can see this is an example of a phytoplankton that we see in our ponds. So again, they're going to form the bottom of our food chain. So these are going to be phytoplanktons that our zooplanktons are going to, or herbivorous zooplanktons are going to feed upon. So the first zooplankton that we see are these rotifers. And these are the smallest zooplanktons that we enumerate. These are extremely important because it's the first zooplankton prey source that the larval fish will be able to consume. Next, we have copepods. So we are classifying them not by species, but we do categorize into copepod nauplii. So these are juvenile copepods compared to the adults. And then we even see some polychaete larvae in our ponds. They're not as numerous as copepods and rotifers, but they can be extremely important, especially in our cobia production. For the purposes of this presentation, I'm only going to focus on copepods and rotifers and really more specific rotifers. And up in here in the top left, you can see a couple of photographs of insects. So these are organisms that we don't have as much control over in terms of entering our ponds. We have limited control with the use of some larval insecticides. However, we do see some dragonfly nymphs entering our ponds on occasion. That can be important because even though they're not in very high numbers, they can be really voracious fish predators. And then we also see caryxids, these water boatmen. They're not predators, they're actually herbivorous, but because of some behavioral mechanisms, they can damage our fish during harvest. They look for vegetation to grasp onto. Sometimes they can grasp onto the gills of our fish and damage them. However, as the fish grow in our ponds, we have actually seen that they consume these, so it can be another part of the prey base. So we do monitor plankton communities in our ponds at a basic level. Here are some phytoplankton species. You can see some diatoms at the top left. We do not classify phytoplankton by species. However, these data are publicly available through an NCOS website. So National Center for Coastal and Ocean Science. They have a citizen scientist volunteer network. Really, primarily, this was instigated in order to look at harmful algal bloom species. However, they do train their volunteers to identify multiple species, harmful and non-harmful, of phytoplanktons. And I've plotted here, these are just the top 6 phytoplanktons that we see here at Waddell. We actually have a couple of volunteers that have been sampling the Waddell dock since 2006. So the important thing to note here is then when I plotted on the y-axis, you can see this is the number of cells per liter of these phytoplankton, and I've plotted that by month from January to December, and there's no difference by month. So this is good news. This means that we aren't limited at the bottom of our food chain by primary producers. So in terms of how we monitor our phytoplankton, it's just very rudimentary. We're looking at color and we're looking at turbidity or clarity of the water. So that's going to give us an indication of how many phytoplanktons we have in our ponds. So the more clear the water, the less phytoplankton we have, and that's going to inform how we fertilize our ponds. So we do fertilize with organic slow release fertilizers like cottonseed meal and alfalfa. We can add urea as an immediate nitrogen source if need be. Our zooplankton, we start sampling usually around 7 days after we start the pond. The ponds are deep enough then to sample. And we do that 2 to 3 times a week. We do that prior to stocking and several weeks after stocking to ensure that we have enough prey base for the fish. You can see here we use this vertical plankton sampler. So we take a 3-liter sample, we condense that down through a 40 micron sieve, and then we take 3 samples from that 3 single milliliter samples, and we enumerate and classify to the lowest reasonable taxa our prey groups. We also take water quality twice a day with a YSI Pro instrument. We take water temperature, dissolved oxygen, pH, and salinity. So here I have plotted the number of rotifers per liter, number of copepods per liter at the top in blue. On the x-axis is pond age. So pond age day 1 is the day that we start filling the ponds. So you can see here, if you look at this bottom graph, just notice here that our rotifers, our number of rotifers are really peaking between day 10 and day 14. So that's pond age. We really want to strive to introduce our fish larvae to the ponds during that period to make sure that they have sufficient prey base. So our larvae are 2 days old when we stock them, and they're ready to start feeding on day 3. So we want to make sure that we have plentiful rotifers for them to eat. The rotifers are more important at this stage because, again, they're small enough for the fish to consume. These are 2 to 3 millimeter larvae, and they're limited by their mouth gape. So this is actually a spotted sea trout, but you can see here, extremely small organisms. They're also not that motile yet. Their fins aren't well developed, so they can't effectively chase a larger, faster predator like a copepod. You can see the copepods are going to start blooming up really around day 18 and 20. So that becomes more important as the fish grows. So our red drum production cycle really begins in our broodstock tanks. So we have adult fish that we have genotyped. So we know genetically we have very unique families. That becomes important so that we can effectively answer our research questions year to year when we release our stocked fish. We condition through a combination of photoperiod and temperature that mimics what's happening in the wild. So our wild red drum are spawning in the August to September timeframe, and we can induce our red drum to spawn at the same time in our tanks through this temperature and light manipulation. We collect the eggs, we place them into hatching tanks, and after about 24 hours, the eggs hatch. Again, at 2 days post-hatch is when we're introducing these fish larvae into our ponds. They're growing out into our ponds late August to November timeframe, generally in our ponds for 30 to 45 days, depending on growth. They need to be at least 30 millimeters in size, about 1 inch in size before we can safely harvest them. After release, at about 10 months old is when they become available to the surveys that Joey was speaking about, in particular electrofishing and trammel netting. And that's important because our inshore fisheries group can take a small non-lethal piece of fin tissue, and our genetics section can analyze that. And then we can determine our hatchery contribution. So we know the difference between wild and stocked fish. So here's a photo of a pond at the time of harvest. So after we introduce the fish into the ponds, we begin to sample using either a plankton sled or a seine net. And we sample at least weekly. And in this way, we can look at the growth of the fish over their time in the ponds. And we can also predict the time that we'll be able to harvest them, the time that it will take to get them to that 30 millimeter mark on average. So all of the fish are collected into this catch basin. We pick them up with dip nets carefully. We place them all into these buckets that have 10 kilograms of water. Everything gets weighed, and then we count small samples, so 100 to 300 grams of fish. We're counting those individually so that we can extrapolate based on our total biomass how many fish we harvest from each pond. So just to let you know, this is one of our larger ponds, so it's 1.25 acres. We're stocking 1 million fish per hectare. So in a pond this size, we stock about 500,000 red drum larvae. And our medium-sized ponds, we're stocking 250,000 red drum larvae per pond. So after we harvest the ponds, all the fish are transferred into our special hauling boxes where they are transported along the coast from Winyah to Port Royal Sound, depending again on our research questions in that particular year. They're either released directly at a boat landing, or sometimes they have a secondary transport into a boat where they are released directly into marsh grass. So for the purposes of this study, I just looked at our 2019, 2020, 2021 production of red drum. So I've broken everything down, not only by year, but you'll see early and late here. So this is an indication of when we stocked fish into our ponds. So again, we begin stocking in late August. So the early time period is stocking from August to mid-September. The late time period is our late September to early October stocking into our ponds. So fish age is just the number of days that they're in the pond +2, because we stock at 2 days post-hatch. This is the total length, the mean total length in the range of total lengths that we're harvesting in the ponds. The overall survival, again, a range of our survivals that we're seeing in those timeframes. And then this is the total number of red drum that were released. So if we looked first at survival by temperature, you can see that we have two really distinct groups that fall out depending on if we stocked fish early in the season. So those are experiencing significantly higher temperatures, about 30 degrees Celsius mean temperature over the life cycle of the pond. But the fish stocked later, really only about 20 degrees Celsius as a mean. So even though the temperatures are significantly different, we didn't see a difference in survival. And then if we look at survival by rotifer density, so I'm going to clarify here that the rotifer density that I looked at was only in the first 10 days after we stocked the fish. Again, this is the most critical period for our rotifer density because our fish are small enough where they can't eat any other zooplankton. So they're not, copepods aren't available to their mouth gape at this point. It's really strictly limited to rotifers. So although we didn't see a relationship between density of rotifers in ponds and overall survival, we did see a significant difference in the density of rotifers in early versus late stocked ponds. So the ponds that we started and stocked later in the season had significantly lower numbers of rotifers. However, this had no effect on survival. What we did see a difference in are growth rates. So if you look at the x-axis, this is total length and age in days. So this is the amount of time that it takes those fish to attain our harvest size of, again, about 30 to 50 millimeters is ideal. So you can see that in orange here, these are our late stocked fish. So it takes them much longer to attain that size that we need them to be at harvest. There's also, so not only do the early stocked fish have the opportunity to get larger, but there's also much more variation in the size at harvest. This is expected because, of course, temperature is going to lower metabolic rate. So we expect that those fish stocked later would grow more slowly. So take-home message from this slide is that bigger isn't always better. So you can see here, again, x-axis is total length, and right here, survival. We are attaining our highest survivals in our ponds when those fish are between about 30 and 50 millimeters. So that's when we want to gear our harvest. If you look at the x-axis and look at this grouping of ponds here where we have fish that are significantly larger, the survival is low. So that can be for a couple of reasons. And this is really why we want to limit the fish, the time spent in the ponds. So I mentioned that we can have a much higher density of zooplankton compared to the estuary because we're feeding and fertilizing the ponds. We're concentrating that prey base. However, as the fish grow, they're going to feed on the zooplankton. At a certain point, we're going to lose the balance and we're going to have a strong decrease in our prey base. Although we supplement with commercial feed, it really isn't as effective at keeping up with fish growth while keeping a high survival. So we could have fewer fish as a result of just losing that prey base. They can also begin to cannibalize each other. This can also be an indication that we had an early mortality event, which is really tricky to actually visualize in the pond. So maybe the prey base wasn't limited, but we had a real early mortality event. The fish had plenty to eat, but there were just far fewer. So in conclusion, we found no effects of survival based on either temperature or prey base. Where we did see significance was in growth rates. So we know that fish stocked later in the growth season are going to they're going to grow slower. But we don't know, is that a temperature effect solely, or does it have, is it based on the fact that we are seeing those lower rotifer densities in the ponds at that time? And this is something really difficult to discern when we're looking at the ponds themselves, but we can begin to tease these effects out by running challenge experiments in our hatchery. And in the future, we'd really like to use some multivariate analysis to assess the importance of not only temperature and prey base, but also other physical and biological parameters like dissolved oxygen, pH, and their interactions in order to look at survival and growth. That helps us not just in terms of making our pond production more efficient, but it can also inform us what's potentially happening with year classes of red drum in the estuaries themselves. And with that, I'd like to acknowledge everybody in the estuarine finfish section. So again, you've heard from Joey and Elizabeth in our inshore fisheries section, and also our genetics group. We really work together as a team to design our research questions in order to best inform what's happening with the population trends of recreational finfish in the state. Also, just a special mention to Matthew Brown, who is our trade specialist here. This is a pretty intense facility to keep running, and he's been here for over 30 years helping us get this research done. And with that, I'll take any questions if there's time.
All right, thank you very much, Erin. The connection here between what is required for larval and juvenile red drum to mature to be the size that they can be caught in the surveys that Joey described, I think is informative. Thinking about our tidal creeks as sources of food and the salt marshes as places for these small larval fish to feed, the more we can learn about this system, the better. With the red drum, you did mention that they cannibalize. Can you give me an idea about, like, if you have you said you would like to release them at 30 to 50 millimeters. What eats a what size red drum eats a 50 millimeter juvenile red drum? You talking about the cannibalism? Something twice its length or?
Interspecific. So we've seen, they'll try to eat something the same size as they are when it gets to that point. So, and it's really when the prey base becomes really depleted. Well, we generally see more of that also with higher temperatures. So the ponds that are stocked earlier.
Okay. So this relates back to what Joey was discussing, was the whole idea of the role of predation affecting population changes we see in when we're trying to do long-term monitoring of Port Royal Sound fish populations. So very good. All right, well, let's move on. Thank you again, Erin. Gives us a lot of insight into what happens with the smallest size that aren't typically measured by other means.